This demonstration shows sodium and glucose cotransport using the SGLT1 carrier protein, which moves sodium down its concentration gradient, carrying a glucose with each sodium ion. The concentration of sodium is high and glucose is low in the gut lumen. In the epithelial cell, concentration of sodium is low, potassium is high, and glucose is high. In the extracellular fluid, the concentration of sodium is high and potassium is low. First, sodium binds to the carrier SGLT protein carrier. The binding creates a site for a glucose. The glucose binds and changes conformation. The sodium and glucose are now released into the epithelial cell. Glucose is a hydrophilic molecule, so it uses facilitated diffusion or transport to leave the epithelial cell and go into the extracellular region. Sodium is then actively transported out of the cell using the sodium-potassium pump. Sodium binds to the pump. 3 sodiums bind to the pump, and this binding stimulates phosphorylation by ATP. Phosphorylation causes the proteins to change shape, and this releases sodium into the extracellular region. The new shape has a higher affinity for potassium. 2 potassiums bind to the sodium-potassium pump, and this triggers the release of a phosphate group, which changes the shape and allows potassium to go into the epithelial cell. This cycle repeats continuously. The ATP of the sodium-potassium pump indirectly provides energy for the cotransport. Water diffuses through aquaporins and channel proteins from the gut lumen to the epithelial cell and then into the extracellular fluid because the epithelial cell is in a hypotonic solution. This is the sodium-glucose cotransport.